This protocol describes a complete purification procedure for the human calcium-binding protein S100A12, and in particular for its different ion-induced oligomers for downstream research applications.
With this technique, we are able to maintain high yield of very pure and endotoxin-free protein. Chemical cross-linking and separation on a size exclusion chromatography column is a convincing method to generate homo-oligomeric proteins for further functional assays.
Our technique allows to study mechanistic aspects of S100A12 as pattern recognition receptor ligand in autoinflammation to ideally develop new targeted interventions and improve diagnostic assays.
In addition to watching this video, we recommend a careful reading of the protocol as we describe a complex multi-step procedure.
To begin, cut a dialysis tubing into an appropriate length around 30 centimeters with additional space for air. Soak the membrane in deionized water for 15 to 30 minutes to remove glycerol. To reduce the viscosity of the cleared protein solution, dilute the solution with 25 milliliters of AIEX buffer A. Attach the first closure onto the tubing. Load the sample into the membrane and attach the second closure at least 1 centimeter from the top end of the tubing. In a cold room at 4 degrees Celsius, place the container with AIEX buffer A on a stir plate, add a stir bar, and the membrane filled with protein solution.